We will start our webinar. I want to thank you and welcome all of you to the virtual International Conference of Environmental Ergonomics webinars that we have been organizing since April 2020. This is now season 3. And again, thanks to my co-organizers, Tadej Debevic from University of Ljubljana in Slovenia, and also Chris Tyler from University of Roehampton in the UK. And I am Stephen Chung, and I'm from Brock University in Niagara region of Canada. So again, we want to welcome all of you to what I think will be a really fascinating discussion on a topic that, if you saw my Twitter post, this was something that I hadn't really thought about, and I think this is going to open a lot of our eyes and minds to a new area or a really interesting area. So I will be sharing my screen. All right, today, can you see my screen now or?
No, doesn't look like it.
All right, just. All right, this should do it right today.
Yeah, it's fine.
All right, so again, what we're going to be talking about today is the issue of patent foramen ovoles and how they may impact environmental physiology. So before we get to that, I will again remind you that this is the virtual component of our International Conference of Environmental Ergonomics, and we had to cancel it last year thanks to COVID, but we are live and rolling for this September, and it's going to be here in my home region of Niagara Falls in Canada, and it will be September 6th to 10th, so right after Labor Day for those who observe it. And the host venue and hotel, you can see, is the Marriott. That is actually not a Photoshop. It is directly overlooking the Canadian Horseshoe Falls. And we will be having all of the info that is arising on ICEE2022.com. So that website is live now, and there is links to registration, to abstract submission, and also to hotel bookings. And there will be more links for things such as the kind of airport shuttles and kind of attractions and stuff, etc., coming up. But any questions or any queries about anything, please email me and our organizing team at ICEE2022Niagara@gmail.com. So as I've mentioned at the start, we have been organizing this since April 2020, and we've had, as the title suggests, a really broad range of environments, everything from pollution to heat to cold, high altitude, pre-cooling, and cold water swimming, some occupational work on firefighting health. That was season 1. And then season 2, we've carried that onwards to topics such as breath holding, to brown adipose tissue, and to prepping for heat in Tokyo, and also with COVID-19 and heat stress in healthcare workers. So this has been the talk so far in season 3, and all of the sessions are available on the Environmental Ergonomics YouTube channel from season 1 onwards, along with science communication videos that I've been posting every Wednesday. So feel free to check those out if you have missed any of them. So with that, I will get to today's talk. Again, it's going to be from Dr. Andrew Lovering from University of Oregon, and also Karleigh Bradbury from Andrew's lab. And they will be talking about, Andrew will give us an overview of patent foramen ovoles and the whole concept of how it affects environmental physiology. And then Karleigh is going to transition more towards a talk on its effect on thermal physiology. As always, you can interact with us with the Q&A tool. After each talk, we'll have a moderated Q&A session before we move to the next talk. So with that, I will stop sharing my screen. And Andrew, you are welcome to take it away.
Okay, thank you. Here. How does that look?
It's good on our end.
Yeah, things are good. Okay, great. Well, I got to thank Stephen, Tade, and Chris for the invitation to speak today. I'm super excited to share some of the work that we've been doing on patent foraminal valleys and how it might be related to environmental physiology. I started off with a picture. I hope you can see my, I'll switch over here to laser pointer, but started off with a picture of the area where we live here in the Pacific Northwest part of the United States. And you can see here the Eugene, the little town we live in, is probably tucked away, hidden somewhere behind these clouds a good chunk of the year. But in the last 15 years that I've lived here in the Pacific Northwest, the climate has changed such that at any given time of the spring or summer, these clouds might actually be replaced by smoke as things are changing here, which I think highlights the importance of the work that environmental physiologists are doing. And I believe that this will become more important as things continue to change. But I'm not here to convince you that the climate's changing. I'm here to get you thinking about a small hole that is near and dear to my heart and possibly near and dear in your heart. Every single one of us at some point has a hole between the two atria of the heart, and it's called the foraminal valley. It's part of the normal fetal development, and without it, you wouldn't survive this normal process. However, 1 in 3 people watching these seminars today has that hole present in adulthood. So there's about a 30 to 40% prevalence of PFO in the general population. So quite a few people have it. It is a source of shunt, and as you can see from the arrows here, that blood flow can go from the left side of the heart to the right side of the heart, or from the right side of the heart to the left side of the heart. I will say, however, it's less of a hole than it is a tunnel, and the shape or design of the hole or tunnel is such that when the left atrial pressure is greater than the right atrial pressure, it pushes the flap or the tunnel closed. So it's a little more difficult for blood to flow from left to right than it is from right to left. But again, it's going to always depend on the pressure gradients across that. So if right atrial pressure is higher, blood can flow right to left. If left atrial pressure is higher, then it's possible that the blood can flow that way. Although the design of the tunnel usually limits that. So in addition to being a source of shunt, it's also a source of arterialization, or a pathway for arterialization of bubbles and blood clots. And for bubbles, this can be problematic if bubbles cross over the PFO into the systemic circulation. If those bubbles can go somewhere like the brain, or they can cause neurological DCS, or neurological sequelae secondary to scuba diving. So some people who have problems due to their PFO, who are scuba divers, will actually get this hole closed, or the tunnel closed. And I'll show you a video later of how this happens and how we're using this to study the impact of the PFO on some of the measures that we do in the lab. It also allows for the arterialization of blood clots. And if a blood clot gets through this hole, it can go to the brain and cause a transient ischemic attack, or TIA, or even a stroke. So a lot of people that know that they have a PFO have had some kind of neurological, negative neurological outcome related to bubbles or blood clots. Most people don't know that they have a PFO. A PFO also allows for arterialization of bioactive substances, and probably the most relevant one to today's talk is serotonin, which is typically metabolized by the lung. But if it bypasses the lung and travels from the right side to the left side, it can arterialize this bioactive substance. And there's a well-known association with elevated levels of circulating serotonin and migraine with aura in people who have a PFO. Now, as I mentioned, most people don't know that they have a PFO, but a lot of people that come into my lab find out whether or not they have a PFO. And we use a technique called saline contrast echocardiography, where we obtain a 4-chamber apical view shown here with the right ventricle, the left ventricle, the right atrium, and the left atrium. And then we inject saline contrast bubbles into a peripheral vein where they'll travel to the right side of the heart. And you can see from this still image here, a lot of bubbles over here in the right side of the heart. And then we count heartbeats to see how long it takes for the bubbles to traverse from the right side of the heart to the left side of the heart. And if they can get there relatively quickly within 3 to 5 cardiac cycles, we typically would call this a PFO. But if it takes longer than 5 cardiac cycles, then we would say those are traveling through intrapulmonary shunt pathways and not a PFO. As I mentioned, whether or not blood will flow from the right side of the heart to the left side of the heart depends on the pressure gradients across the intraatrial septum. So under normal conditions, the left atrial pressure shown here, LAP, is higher than the right atrial pressure. And so you can see microbubbles floating around in the right side of the heart, not being able to get across the PFO. However, during spontaneous breathing, at the end of a normal inspiration, during diastole, these two pressure gradients will switch, shown here, and bubbles can flow from the right side of the heart to the left side of the heart. Now, if you're an awesome, super fantastic researcher and experimenter, you can inject the bubbles in somebody and they'll appear in the heart right at the end of a normal inspiration during diastole. But if you're like me, you usually don't have that kind of luck. So we have to have subjects perform what's called a Valsalva maneuver, which is where subjects close the upper airway, bear down really hard for about 10 seconds. We wait for the bubbles to arrive in the right side of the heart. We have them release the Valsalva. There's a transient increase in venous return, and it increases the right heart pressure and will push those bubbles across the PFO. And I'm going to show you some videos here of what this actually looks like. So this is a video of a subject who has a PFO. You're going to see bubbles appearing in the left side of, in the right side of the heart here. And then within 2 to 3 cardiac cycles, you'll see bubbles pop over into the left side of the heart. And this is one of the examples where we got lucky and injected bubbles at the right time. So there's the heart beating, hopefully. See bubbles appear, and then 1, heartbeat 2 bubbles, heartbeat 3 bubbles. And so we can see bubbles jumping over across the PFO. And you see it doesn't happen every heartbeat. It doesn't happen every cardiac cycle. It only happens when the pressure gradients allow for the blood to flow from the right side to the left side. So same subject now performing a Valsalva maneuver. You're going to see the same thing, bubbles appearing in the right side of the heart. But when the subject goes to release the Valsalva, you'll see a large number of bubbles go from the right side of the heart to the left side of the heart. So here's the subject performing the Valsalva. Bubbles appear, release, and then a larger number of bubbles getting across, because we've basically created conditions where at the same time we're imaging, we've allowed a transient increase in right heart pressure to open up to allow the blood to flow from the right side to the left side. What I don't want you to leave today thinking is that you have to do a Valsalva maneuver to open the PFO. That's not true. We only do it so that when we're imaging, we can get the best conditions to see what's the possibility of bubbles getting across that PFO. So I actually started my PFO journey doing something completely different from PFO. I actually started working with this gentleman here, Jerry Dempsey, looking at pulmonary gas exchange efficiency at rest and during exercise, and trying to figure out whether or not intrapulmonary shunts were involved in this, and whether or not these intrapulmonary shunts could be used to explain some of the variability in pulmonary gas exchange efficiency that we see in across different people. So some people have worse pulmonary gas exchange efficiency than others, and we were studying intrapulmonary shunts initially. Well, in order to do that, you have to exclude people that have intracardiac shunts. So we learned how to screen for PFO so we could exclude these people from our studies. But after several years of excluding lots of potentially good subjects, we decided we might want to look at the impact or the possibility that intracardiac shunts were also playing a role in some of this variability. And in our first study that we published in 2011, we found that pulmonary gas exchange efficiency was worse at rest only, not during exercise, in people with PFO. And this was true in both normoxia and normobaric hypoxia. And this was kind of interesting to us because we also, it was confusing that it was causing a problem under resting conditions, but not exercise. And then it was also, we also found that at maximal exercise, the subjects that had the PFO also had lower arterial oxygen saturation, which really didn't make any sense initially because they didn't have significantly lower PaO2. So the arterial partial pressure of oxygen was about the same in both groups. However, we did stumble upon this interesting observation that their esophageal temperatures were higher in the PFO subjects compared to the PFO negative or the subjects without PFO. And that was a really kind of a totally surprising finding. And full disclosure, the only reason we measured esophageal temperature at that time was so that we could correct our blood gas values. We really didn't care what their temperatures were. We just needed to know what they were so that we could correct our blood gas values. So the whole temperature story that Karleigh's going to follow up on today after my talk, it came out of an incidental finding, something that we weren't even initially looking for. So if there's anything that the trainees take away from this is pay attention to everything that you learn in your studies, and you may find something cool to do later on. Now, two of the big limitations in this 2011 study was that we didn't consider the size of the PFO or the volume of blood that could travel through that PFO. So that was one of the limitations. The second limitation is that we didn't have a good distribution of men and women between the two groups. So knowing that, we had more work to do, obviously. Right about that time, a friend and colleague of mine, Brett Fenster in Colorado, was doing studies in people with PFO using an intracardiac echo probe, or intravascular ultrasound. So here, Brett would place an ultrasound probe via the femoral vein into the right side of the heart. So you can see this is an ultrasound view from inside the right heart, right atrium here, left atrium here. And you can see this little line of color here showing the tunnel of the PFO. And this is a small PFO here. And then Brett would inject bubbles into a peripheral vein like we do, like anybody looking for PFO does. And you can see that there were only a few microbubbles getting through these small PFOs. Similarly, in individuals that had a larger PFO, so here you can see a larger tunnel on the ultrasound. Again, inject bubbles into the right atrium, and then see a much larger cloud of bubbles, similar to what you just saw in our video. So adapting it, and Brett found a relatively good association with the number of bubbles getting from the right side of the left side based on the size of the PFO. So we had our own scoring system at the time. So obviously, if there are no bubbles getting over to the left side of the heart, these people have no PFO. If they have 1 to 3 or 3 to 12, or basically anywhere from 1 to 12 bubbles getting across in any frame, we would call that a small PFO. And then if they have a large PFO, that means they've got greater than 12 bubbles in any frame, and a bolus, heterogeneously distributed or homogeneously distributed in the left side of the heart. So these are large PFO, small PFO. I cannot tell you the exact size of the PFO. I just know if it's large or small, or what we would say insignificant or a significant source of shunt. So now we fast forward to 11 years to 2022, and my graduate student, Tyler Kelly, is following up on these studies, looking at pulmonary gas exchange efficiency at rest and during exercise in both men and women in people with large and small PFOs. And what Tyler has found, so we use the alveolar to arterial oxygen difference to quantify pulmonary gas exchange efficiency. You can see that it increases in everybody during exercise. So basically the AADO2 will increase, meaning that pulmonary gas exchange efficiency is getting worse. However, what we're finding now is that in the women with large PFO, so that's PFO plus. So in women with large PFO, gas exchange efficiency seems to be worse than any of the other groups. And that would be male with a large PFO here in green, or females and males in the blue and red with no PFO. So we don't exactly know why the large PFO only impacts the women, but we believe it has to do with the differences in the work of breathing between men and women, such that women have to have greater intrathoracic pressure swings for a given level of ventilation, and that might be facilitating blood flow across the PFO. But we have some measures to do with JJ Duke coming up to look into this to see if that's actually the case. Now, at this point, everything I've shown you is just simply association data with the gas exchange. If these people with a PFO, people without a PFO have better or worse gas exchange. And as I mentioned, there are individuals who sometimes have negative things happen associated with this PFO. And I'm going to show you a video here as an example of that. So you've got here you can see blood clots coming to the right side of the heart. You can see the blood clots in the atrium here. You'll see one of them gets a little too close to the PFO and is able to sneak across into the arterial side, where it is becomes arterialized, gets pumped out into the systemic arterial circulation. And in this case, unfortunately, it travels to the brain, where it becomes lodged and can cause a transient ischemic attack or a stroke. Now, the cardiologists in our area and all over the world can close this hole with this little device you're going to see here on a right heart catheter. It's a little mesh disc that comes out of the catheter, goes downstream here. They pull the catheter back. The other side of the little mesh cap comes out here, and they connect in the little tunnel. And then after 3 to 6 months, cells will grow over this, and it will endothelialize and become part of the interatrial septum, so that the next time a blood clot or a bubble comes by, it hits this and doesn't go through, and everything is good for these individuals. So working with the local cardiologists in our group, we're now studying, Tyler, my grad student Tyler, is studying people pre-closure and post-PFO closure. So here again, we have the alveolar to arterial oxygen difference, which shows the pulmonary gas exchange efficiency. And you can see pre-closure in blue, the AADO2 is somewhere around 10, and then with exercise increases up to close to 30 millimeters of mercury. However, post-PFO closure, we see a substantial reduction in the AADO2 or an improvement in pulmonary gas exchange efficiency. And I will point out that there are error bars here. The numbers are just so tight you can't see them. So we believe that the PFO is a significant source of some of the variability in the AADO2s that we see, and it probably should be taken into account in people doing pulmonary gas exchange efficiency or anything related to arterial oxygenation moving forward. Now, right around the time we were publishing our first paper on the PFO and gas exchange, we had an opportunity to take our show on the road, so to speak, and traveled to Bolivia with Rob Roach and Andy Sabuti and Colleen Julian as part of the Altitude Omics expedition. And so here you can see a group of the researchers walking back from the little research station here on Mount Chacaltaya, and we're looking back at Huayna Potosí, which is a 6,000 meter peak just outside of La Paz. And we took people with PFO and people without PFO in hopes of learning something about pulmonary gas exchange efficiency, which we did. And I won't talk much about that today, but I'm happy to answer any questions about what we found. But one of the interesting things or surprising things that we found on that trip was that the people with PFO were the ones most likely to develop acute mountain sickness on the first day, and even still have symptoms of acute mountain sickness as many days out as day 5. Whereas the people without a PFO were less likely to get AMS, and they didn't have any AMS symptoms as of day 5. And of course, after being on the mountain for 16 days, they were both just fine. So we first reported that in Journal of Applied in 2015, and more recently, Brian West in Jonathan Tobus's lab did a field study looking at individuals hiking above 3,000 meters on the John Muir Trail, and found that if you have a PFO, you were more likely to have AMS on your hike above 3,000 meters, even if you were taking drugs for AMS prophylaxis. And so we've combined these two field studies, the one done in our lab and the one done by Brian West, to show that the odds ratio of developing acute mountain sickness if you have a PFO is 2.88. So here we have our AMS negative subjects. PFO negative is in red, PFO positive is in blue, and here AMS positive subjects, PFO positive again in blue, PFO minus in red. So following up on these studies, more recently in our lab, Caitlin DeMarco, one of the grad students in my lab, took a bunch of subjects and exposed them to hypobaric hypoxia for about 10 hours, 11.5% oxygen. So this is normal baric hypoxia. And we didn't find the same association in PFO positive subjects. So whatever's causing this, it's hypobaric hypoxia only. And so for the trainees out there trying to get their advisors to fund more field research, you can use this and tell them like, look, the normal baric hypoxia is not going to work. We have to go out, we have to go to the mountains. And that's true only of your students, not of my students. So the real question now is, what is it about a PFO that's making things bad for people at altitude? So the question, the usual suspects that people look at when we talk about AMS, we think about arterial oxygen saturation, we think about cerebral vascular reactivity, we think about hypoxic ventilatory responses. And so we've looked at all those things, and I hope I can convince you today that I don't think it's any of those things for the PFO. So this study done by JJ Duke, I think there's a picture, I thought there was a picture of JJ there, so I apologize. So this study done by JJ Duke in the lab had subjects breathe 21% oxygen, 16, 14, 12, and 10% oxygen in a random fashion for 30 minutes. And we measured pulmonary gas exchange efficiency, AADO2 again, between the PFO negative subjects, which are the filled circles, and the PFO positive subjects, which are the open circles. And we found at the moderate altitudes or in room air that there were significant differences in the AADO2. However, as you get to the more severe levels of hypoxia, and this little arrow here is to show that this is right around the same, would be similar to the same altitude as Mount Chakotay at 5,260 meters. That what causes differences in AADO2 at more severe levels of hypoxia or more severe altitudes is a diffusion limitation and not sources of shunt or ventilation perfusion mismatch or things like this. Because the higher you go, the more likely your venous, sorry, I have to say this again, the higher you go, the closer the partial pressure of oxygen from the arterial blood gets to the mixed venous blood. And said another way, basically at sea level, your arterial blood has a partial pressure somewhere around 100 millimeters of mercury, and your mixed venous blood has a partial pressure somewhere around 40 millimeters of mercury. So any mixture of a low PO2 with a high PO2 has a large impact on the gas exchange efficiency. But as you start going to higher altitude, your arterial PO2 starts to get much closer to your mixed venous PO2, so that the sources of shunt have a minimal impact on gas exchange efficiency. And in this study and in the Bolivia study, we find no differences in arterial oxygen saturation in these individuals between PFO positive and PFO negative. So it's hard to attribute it to that. One of the things we did find that was interesting was that after 16 days of being at 5,260 meters, those people with a PFO had blunted ventilatory acclimatization. We know that because they had a lower P(a)O2 and a higher P(a)CO2 than people without a PFO. So they increased their breathing, they just didn't increase their breathing as much. So they did have lower saturations after 16 days. The problem here is that at 16 days, nobody had acute mountain sickness. So there's a long-term effect of having a PFO on gas exchange efficiency and saturation, but for the short term, there's just no detectable effect. And we found that also true with hypoxic ventilatory response. In 2015, there were no differences between the two groups with acute ascent to altitude. And then we followed up with these studies, Jim Davis in my lab and Lindsay Boulay working with Glenn Foster at UBCO, followed up these studies a few years later and tested a significantly larger group of subjects with more men and more women. And we found that there were no significant differences in acute hypoxic ventilatory responses with either poikilocapnic or isocapnic hypoxia. And then last but not least, Mike Timko working in Glenn Foster's lab showed that there's normal cerebral vascular responses to low oxygen and high CO2 in people with a PFO. So it doesn't appear to be related to saturation. It doesn't appear to be related to chemosensitivity or cerebral vascular reactivity. So we're trying to start moving beyond hypoxemia in my lab. And my grad student, Caitlin DeMarco, shown here, is working on a series of studies looking at circulating cytokines in people with and without PFO. And here you can see higher levels of circulating cytokines in people with PFO and the gray bars here. And excitingly enough for us, in those individuals undergoing PFO closure, we see significant reductions in circulating cytokines following PFO closure, which we think is moving these studies beyond just association and more along the lines of causation. Now, acute mount sickness isn't the only thing that's associated with the PFO. The PFO is also associated with high altitude pulmonary edema, or HAPE. And this was first reported by Alleman in JAMA in 2006. So what they found in these individuals is a larger or a higher prevalence of PFO in those individuals who are HAPE susceptibles. And one of the hallmark responses of individuals who are HAPE susceptibles is an exaggerated hypoxic pulmonary vasoconstriction that you can see here. Compared to controls, these individuals breathed 12.5% oxygen for 4 hours, and pulmonary pressure increases with exposure to hypoxia. We know this. However, in those who go on to develop high altitude pulmonary edema, they have these exaggerated pulmonary vascular responses and a much greater increase in pulmonary pressures. So we wanted to know, if you have a PFO, does that predispose you to having an exaggerated hypoxic pulmonary vasoconstriction? And I'm sad to say that the answer is no, it does not. So here's the picture of JJ I was looking for earlier. So here we have subjects without a PFO, subjects with a PFO. JJ had these individuals breathe 21%, 16, 14, 12, and 10% oxygen for up to 30 minutes. You can see it's progressive increase in pulmonary artery systolic pressure in both PFO negative and PFO positive subjects, but no significant differences in the pulmonary pressure responses, save maybe for this individual here who might actually be a HAPE susceptible person, although we don't know that. Kaylin DeMarco extended these findings looking at pulmonary pressures up to 10 hours. So we have baseline, half hour, 1 hour, 4, 7, and 10. This is pulmonary artery systolic pressure, PFO positive, PFO negative. This is the same group, but divided out into the different sexes. No significant effects here. This is cardiac output here, and then total pulmonary resistance, which is pulmonary artery systolic pressure divided by cardiac output. So we see no significant differences between those individuals with and without a PFO. So the take-home message here is, just because you have a PFO, it doesn't mean you're going to get high altitude pulmonary edema. However, if you do have an exaggerated pulmonary hypoxic pulmonary vasoconstriction, which increases your right heart pressure, and you have a PFO, that might allow for more blood to flow across that PFO and cause problems. But we haven't studied individuals that are high altitude pulmonary edema susceptible yet. So as a summary, pulmonary gas exchange efficiency is worse in women with large PFOs, and this is reversed following PFO closure. So we think this is pretty good evidence that the PFO is involved in gas exchange efficiency. PFO is also associated with normal acute hypoxic ventilatory response, but with blunted ventilatory acclimatization to high altitude. So in the short term, we don't see any differences in ventilatory responses, but in the long term, we do see significant differences in their ventilatory responses. We know that PFO is associated with acute mount sickness and HAPE. However, these associations with AMS and HAPE are not necessarily explained by what I would call the usual suspects, which are hypoxemia, and in altered ventilatory responses and cerebrovascular responses. And if I have any time left, I'm not sure exactly where I'm on time. I want to just give some future directions and some acknowledgement. So moving forward, we've got a study planned this summer called the Colorado-Oregon Altitude Study, or COAST 2022. And we're going to take, we're just going to study normal human acclimatization to a moderate altitude with and without some interventions. We plan to take about 100 subjects with, some of them are going to have a PFO, some of them are not going to have a PFO. And we're going to take them from Eugene, Oregon, which is about 131 meters above sea level, to Leadville, Colorado, which is about 3,100 meters above sea level. And we're going to keep them there for 2 weeks and study them acutely, and then over the course of the 2 weeks of acclimatization. The interventions we have planned right now are erythropoietin, intravenous iron, or they'll get a placebo. And then we'll look at some of the usual suspects, AMS, hemoglobin mass, ventilatory acclimatization, some thermoregulatory aspects, but we'll also be looking at some of the non-usual suspects, and those will be circulating levels of cytokines and things like this. So hopefully next time you see us talking, we'll have some COAST 2022 data to present and not 10-year-old data from Altitudeomics in 2012. And then I have to thank all the people that worked on this project over many years. All the individuals that are highlighted in purple here are the ones that contributed to the work that I presented today, but I just wanted you all to see the entire team that was involved in running the studies at altitude and getting everything to market, so to speak. Our subjects have been fantastic, living with us in altitude and letting us put them through the ringer, if you will, by giving them really, really low levels of oxygen and still coming back for more. And then I've got to thank the Department of Defense and the American Heart Association for the funding that we've got to do these studies. And I will close with a picture of a heart that was dissected here at the University of Oregon that was found by my grad student Caitlin, who teaches in the anatomy course here. And you can see the probe here going through the tunnel from the right side of the heart to the left side of the heart. And if you have any questions, I'm happy to take those now.
Great. Thank you very much for that, Andrew. And I will remind the audience, if you wish to ask any questions, please use the Q&A tool and I will pose them on your behalf. I guess I'll start off with, just another thought question. Given the work you've done on kind of hypoxia and stuff, and I know, as you know, Phil Ainsley and their work with Global Reach recently have looked at a lot of natives in Peru with very high red blood cell count and that polycythemia. Like, I guess just can you brainstorm or kind of give us your thoughts on whether PFOs in those individuals might exacerbate any of the findings that they've seen in polycythemic individuals?
Yeah, no, that's a great question. So we've done some work with Phil looking at PFO prevalence in Sherpa. I did some work with Ben Kaiser in China, looking at PFO prevalence in Tibetans compared to Han Chinese. And we don't see any differences in prevalence in those two groups. I will say that there are some studies to suggest that people that have a PFO, people with South American ancestry that have a PFO can develop exaggerated increases in pulmonary pressure during exercise. So there's definitely a possibility that the PFO may be involved in the polycythemia, but as far as I know, I don't think it's been looked at. That's a good question.
Yeah, it just opens up the door, literally, hole in many questions. But I guess just to bring it back away from environmental stress, can you just remind us, in terms of just PFO in the everyday population, are there any kind of major kind of health issues, changes in exercise capacity, prevalence of exercise-induced arterial hypoxemia, like anything like that to set the stage?
Yeah, no, those are really great questions, and I think the short answer is yes. It depends. Yes, but it depends, right? So we think that we know that in women, there's a population of women that develop exercise-induced arterial hypoxemia at lower exercise intensities than men, and we think that the PFO may be contributing to that. And we actually just submitted a grant to do a series of studies like this. So I definitely believe that it's involved there. It might be involved in men who have either obstructive lung disease or an exaggerated work of breathing because they're maybe elite athletes, and so they do have these higher intrathoracic pressure swings. But again, these are questions that we haven't been directly addressed that we hope to address in some upcoming studies. As far as your question about is there anything about the PFO in normal people day-to-day life that has any kind of negative impact? As I would say, the short answer is no, but the longer answer is there is an association, and we know that people that the blood clots can get through the PFO and or the bubbles can get through the PFO, where they will cause a problem. I think it requires a series of unfortunate events. So you have to have bubbles and or clots coming through. They have to be coming back to the right side of the heart at the same time that the PFO happens to be open. And then the blood that's carrying those bubbles or the blood clot also has to go to the brain. Well, we know that 100% of the cardiac output doesn't go to the brain. So you could pass a clot or a bubble and it go to your leg and not cause the same kind of problems that it would if it went to your, I think I said leg and then as it would if it went to your brain. So yeah, I think there are some potential, negative things that can happen, but for the most part, 30% of the population doesn't walk around getting strokes, right? And 30% of the population doesn't have exercise-induced arterial hypoxemia. So I think it has a potential role in those problems, but it needs to be figured out what that exact role is.
Great. And we have a question from Tony Bain, who I'm sure you know.
Yeah.
Great stuff, Andrew. Do you have any speculation on why cytokines are elevated in those with PFO?
Yeah, so that's a great question. The short answer is I don't have any idea of why they would be elevated. We were excited to see that they were decreased following PFO closure, which suggests some role of the PFO in that instead of just simple association. Yeah, I was hoping maybe that Tony could give me the answer because he's already given me so many other answers related to things that I do. I was hoping maybe he was gonna type in the answer to his own question. But yeah, the short answer is we don't know.
All right. Thanks. And today, any questions from you?
Yeah, I just have a short one. I mean, really a great presentation. For me, it was fascinating. I mean, I'm not that much into this topic, but is there, so just as a practical side, is there an easy way to assess if somebody is PFO positive or negative, or this is the strategy that you use is kind of, let's say, the golden standard and the most viable way to test?
Yeah, so there's no easy way. You can't look at somebody and say, oh, that person has a PFO or that person doesn't have a PFO. It's not that easy. There's no simple blood draw. There's no blood test you can take. You have to inject bubbles. And depending on who you talk to, I think the true gold standard is putting in a right heart catheter and watching the catheter go through the tunnel. But you can also use transesophageal ultrasound, where you can put in a esophageal ultrasound probe and get a really good image of the tunnel. Of course, that's not the most comfortable thing for the subjects. We use transthoracic ultrasound because most of the people we work with are relatively healthy. They don't have lung disease, so it's easy to get good ultrasound windows. And you can also use a transcranial Doppler with the microbubble injections to look for TCD hits, because if the bubbles get across the PFO, you can typically pick them up in the cerebral circulation as well. I will tell you from my own experiences when we have done simultaneous TCD and transthoracic echo, that you have to have a pretty high bubble score you have to have a pretty good sized PFO to pick it up with transcranial Doppler. And it's possible you can miss some of the small PFOs with TCD. I'll also say if you're in a place that's hypoxic, hypoxia opens intrapulmonary shunts and allows bubbles to get through the lungs. And if that's true and you inject bubbles in somebody, you don't know if they're getting through an intracardiac shunt or an intrapulmonary shunt. So that's why you'd have to do the transthoracic, where you can actually see the heart to see where the bubbles are coming from, if it's from a PFO or from intrapulmonary shunt.
Yeah. Thanks a lot. So there's no easy solution in a way. I mean, for a general public, just
yeah, you're getting bubbles and some type of ultrasound
Exactly.
or a right heart catheter, which I would go with the ultrasound first.
Yeah, probably.
Great.
Thanks.
Thank you so much, Andrew. We'll move on to the next speaker, and there is a question in the Q&A tool from Fabian Muller that you can answer while Karleigh is speaking. But I'll now introduce Karleigh Bradbury, who's a PhD candidate with Andrew at U of Oregon, and she's going to focus more on the thermoregulatory angle. So first, Andrew, if you can stop your screen share.
do that. Maybe. I think my screen is like frozen here.
Yeah, I will stop that on your behalf.
Thank you.
All right, you can see that okay?
Yes, you can see everything fine.
Awesome. All right, so the title of my talk is Patent Foraminal Valley and Thermophysiology, Are There Holes in the Story? So I'd first just like to thank Stephen, Chris, and today for the opportunity to present some of our PFO thermoregulation research, and thank the rest of you for coming to listen to our talks. So for a quick presentation overview, I'm going to talk about how we even got started studying PFOs and core temperature and thermoregulation, go into some previous studies involving exercise, some passive heating and cooling. That was all work done by a previous grad student in our lab. Talk about some more recent work looking at the heart rate and core temperature relationship between PFO positive and PFO negative subjects, and then finish up talking about our current work and potential future directions. So as Andy mentioned earlier, the PFO core temperature findings really happened on accident. And in that pulmonary gas exchange efficiency study, they collected esophageal temperatures with the sole purpose of being able to correct their arterial blood gases later. So when they're analyzing the results, they found that the PFO positive group had lower arterial oxygen saturations despite having the same arterial PO2s. So when we think about what would cause that, so a lower saturation for the same arterial PO2, or what would cause a rightward shift in our oxyhemoglobin curve, they ended up finding that the PFO positive subjects had higher esophageal temperatures by about 0.4 degrees Celsius. But in that study, they really lacked the controls that we'd usually think about having in place when we're doing core temperature or thermoregulation research, like the control for time of day, hydration status, or even menstrual cycle in our women. So they thought that these findings were really just an accident. So after that initial study, the goal is to figure out if these differences in core temperature in the PFO positive subjects were real. So during exercise, well, we know it's not a primary mechanism for heat loss. We know that we do lose heat through both convective and evaporative cooling, and it accounts for about 10 to 14% of the heat loss during exercise. We also know that the PFO is an intracardiac shunt. So blood that's flowing through the PFO is going to bypass that pulmonary circulation. So there may be differences in respiratory system cooling between PFO positive and PFO negative subjects. And that may be a difference or a mechanism for the differences in core temperatures that we're seeing. So the purpose of this study was threefold. It was first to determine if there are differences in temperature between PFO positive and PFO negative subjects or men at rest and during exercise. And then determine if the difference in core temperature is dependent on the size of the PFO. So like Andy mentioned, we can have small PFOs and large PFOs all dependent on their bubble scores. And third, does breathing cold air, so approximately 2 degrees C versus ambient air, so just room temperature, 20 degrees C, impact core temperature in the same way in both PFO positive and PFO negative men? So the thought behind breathing the cold air is that if the PFO positive subjects are going to have less respiratory system cooling due to that blood being shunted from the right side to the left side, they'll have less blood coming in contact with this cold air in the lungs and may be able to maintain their core temperature. So in this study, they hypothesized that PFO positive subjects would have higher esophageal temperatures compared to PFO negative subjects, both at rest and during exercise. Subjects with large PFOs would have the highest esophageal temperatures at rest and during exercise. And that in response to breathing the cold air, the PFO negative subjects and subjects with a small PFO would not increase esophageal temperature by the same amount as the ambient air trial. So they would have lower core temperatures while breathing the cold air compared to the ambient air. And then subjects with large PFOs would increase esophageal temperature to the same or greater degree compared to the ambient air trial. So in this study, they studied 30 young, healthy men. And on the very first visit to the lab, they underwent an ultrasound screening to determine whether or not they had a PFO. And they ended up studying 15 PFO positive and 15 PFO negative men. They came back to the lab 3 more times. They did a VO2 peak test, which helped us determine our exercise intensities for the exercise workloads for the graded exercise protocols that they would do. And during those graded exercise protocols, they cycled for 4 different workloads for 2.5 minutes apiece. and they were at 25%, 50%, 75%, and 90% of VO2 peak, one day breathing that ambient air and the next day breathing the cold air. We measured esophageal temperature with the esophageal probe. It was all just done in our lab environment, so the average temperature was around 21 degrees C. And in this study, they had in place a control for time of day. So you can see the setup here for the day that they breathe the cold air. So they have this corrugated tube, and in this bucket is a saltwater ice slurry that helps cool down the air that they were breathing in. And in here, they have an esophageal temperature probe, so they can measure the temperature of the water. And then up here, they had another hole drilled into the side, the inspiratory side of the Hans-Rudolph valve, where they could measure the temperature of the air that the subjects were breathing in. And in this ACE bandage is a bunch of ice packs to really make sure the air was cold by the time they got to the mouthpiece. So for some results for this study, this is looking at esophageal temperature during the VO2 peak test. And on our x-axis, we have workload. On our y, we have esophageal temperature. Our PFO negative subjects are in this blue filled in circles, and the PFO positive subjects are in the open red circles. And you can see that at rest and throughout the entire duration of the VO2 max test, the PFO positive subjects had higher core temperatures compared to the PFO negative subjects. And that difference was about 0.4 degrees Celsius. So almost the same difference that they saw initially in the pulmonary gas exchange efficiency study. So they determined, yes, PFO positive subjects do have higher core temperatures. Then they wanted to look at the size effect. So this time, the small PFOs are in these half filled in purple circles, and the large PFOs are in these open circles, the open red circles. And you can see that, well, there's a slightly higher temperature in the small PFO group. There's no significant difference between PFO negative subjects and subjects with a small PFO, but that significant difference is really being driven by the subjects with large PFOs. So when Jim and Andy were thinking about some of the differences of why we could be seeing these differences in core temperature, Jim was able to calculate that about a 2% shunt, or 2% of the cardiac output going from the right side to the left side through the PFO, that could account for about 0.1 degrees Celsius or 25% of the difference in core temperature between these two groups. But about 0.3 degrees Celsius or 75% of the difference can't be explained through the blood being shunted. So it has to be differences in sweating, skin blood flow, maybe metabolism or other factors. So this next slide is looking at respiratory system cooling and esophageal temperature. So in this, it's looking at the difference in temperature between the cold air trial and the ambient air trial in PFO positive and PFO negative across these four workloads. So it's just important to note that in both trials, their temperature did go up across the 4 workloads. It's just the difference in temperature between those 2 trials. So the PFO negative subjects had lower core temperatures at each workload while breathing the ambient or the cold air compared to the ambient air. And that difference was about 0.3 degrees Celsius. But the PFO positive subjects had no difference in core temperature during each workload while they were breathing the cold air compared to the ambient air. When we divide that out and look at the size effect, our PFO subjects with the small PFO, they have slightly colder temperatures while they're breathing the cold air. But for some reason, subjects with a large PFO have warmer core temperatures breathing the cold air compared to the ambient air. So when we think about maybe a potential mechanism that could be contributing to this, we know that when people are breathing cold air, we're going to have peripheral vasoconstriction. Maybe there's an exaggerated vasoconstriction in our large PFO subjects, leading to further decreases in skin blood flow, decreasing heat dissipation, and leading to increases in esophageal temperature. But maybe that's part of it, but we're really not sure of why we're having increases in temperature when people are breathing cold air. The last little bit of data from the study is looking at the effect of a PFO and heart rate. And this is our pre-exercise or resting heart rates between PFO negative and PFO positive. And you can see that there's a higher heart rate by about 10 beats per minute in our PFO positive subjects. There's no difference at any of the workloads at VO2 max or the graded exercise protocol, but at rest, there's higher heart rates. Something else we wanted to point out from this graph is that even though there's a significantly higher heart rate in our PFO positive subjects, you can see the great amount of variability in the heart rates, and we see the same thing in core temperature between the 2 groups. And there's also a really good amount of overlap. So while we're seeing these higher temperatures, it doesn't mean that everybody with the PFO has higher temperatures. The next study that Jim did was looking at the effect of a PFO in humans on thermal responses to passive heating and cooling. And in this study, they looked at 13 PFO positive and 14 PFO negative subjects, and they did a hot tub exposure and a cold tub exposure. So in the hot tub, they stayed in for up to 30 minutes or until their esophageal temperatures reached 39.5. And then in the cold tub, they stayed in there for up to 60 minutes until esophageal temperature got down to 35.5 or until they had sustained shivering. And in this study, we determined sustained shivering by an increase in VO2 by greater than 25% for 5 minutes or more. So this first graph is esophageal temperatures pre-hot tub immersion and end of immersion. And you can see that same thing we saw in the exercise study with our PFO positive subjects having higher temperatures at the beginning of the immersion, throughout the entire immersion to the time they got out of the hot tub. What we think is some of the coolest data to come out of this study is looking at the ventilatory thresholds and the thermal hyperpnea in these two individuals. So on the x-axis, we have esophageal temperature. On the y, we have minute ventilation. And in this solid blue line is our PFO negative group. And you can see that as the PFO negative subjects heat up, they have slight increases in ventilation until they reach a core temperature that's slightly below 38 degrees Celsius. And after they heat up after that, they have a big increase in ventilation. If we look at our PFO positive group, they too have those small increases in ventilation, but they don't have their ventilatory threshold or that big increase in minute ventilation until their core temperature is about 0.7 degrees higher than the PFO negative group. So they don't get to a core temperature until around 38.5, and that's when they really increase their ventilation. After that increase or the ventilatory threshold, even though they're increasing their ventilation, they don't increase it as much as our PFO negative group. And we know that if we look at our end-tidal CO2s. So the end-tidal CO2 in the PFO positive group is 35 millimeters of mercury, but in the PFO negative group, the end-tidal CO2 is all the way down to 28 millimeters of mercury. So PFO negative subjects are breathing much more than our PFO positive subjects. So if we look at our 3 drives to breathe, our PFO positive subjects have higher end-tidal CO2s, higher esophageal temperatures, and lower end-tidal O2s, yet they're breathing less than our PFO negative subjects. Going back to talking about respiratory heat loss between these two groups, in the hot tub study, they were able to measure that. And you can see in the Hans-Rudolph valve, they had vICEE probes in the inspiratory side and the expiratory side, and that was able to measure both inspiratory and expiratory temperature and humidity. And then if we take into account minute ventilation, we can measure total respiratory heat loss in watts. So you can see pre-immersion, there's no difference in total respiratory heat loss, and there's no difference in minute ventilation. But if we look at end of immersion, even though it's not significantly different, there's slightly higher amounts of respiratory heat loss in our PFO negative group, but that's really being driven by the significantly higher minute ventilations that we also saw in the last slide. So per breath, both groups are able to lose the same amount of heat. The PFO negative subjects are just breathing more than the PFO positive subjects. Moving into some of the passive cooling data. So we have our shiverers on the left and our non-shiverers on the right. And we can see that regardless of whether or not PFO positive subjects shivered, they're able to maintain higher core temperatures throughout the cold tub immersion compared to our PFO negative subjects. And you can also see that they start shivering at higher esophageal temperatures compared to PFO negative subjects. So it's almost like something about having a PFO or our PFO positive subjects, they want to maintain higher core temperatures compared to our PFO negative subjects. So going back to that initial exercise study and looking at the differences in heart rate and core temperature, we know that both active and passive heat stress are going to lead to increases in core temperature. And we also know that in a given individual, as core temperature increases, their heart rate's also going to increase. We know that's due to 2 mechanisms. We have the direct effect of temperature on our SA node. So as that heats up, it's going to increase its firing rate and increase heart rate through that mechanism. And then we also have the activation of the sympathetic nervous system, increasing norepi release and increasing heart rate. We also know there's biological variability in core temperature. So if any 2 of us were to go to the lab on a given day, we're not going to have the same exact temperatures. What we don't know is in a group of individuals, are these differences in core temperature associated with differences in heart rate at rest and during exercise? And does the higher esophageal temperature in PFO positive subjects influence heart rate responses during exercise? And third, is there a potential for an altered cardiovascular and thermoregulatory relationship in our PFO positive subjects? So the first thing we wanted to do was look at our whole group of subjects. So this is the same 30 subjects we looked at in Jim's first study and determine whether or not there's a significant relationship between core temp and heart rate at rest. And you can see that there is. So those who came to the lab with lower core temps, around 36 degrees Celsius, they had the lower heart rates, whereas those who came to the lab with warmer core temps, around 37 degrees Celsius, they had the higher heart rates. When we divide that out into PFO positive and PFO negative, you can see that there's no relationship between core temperature and heart rate in our PFO negative group, but that relationship's really being driven by our PFO positive subjects. When we look at some exercise data, we can see the same exact thing we saw pre-exercise, where in our whole group, we have a positive and significant relationship between PFO positive or between core temp and heart rate. When we divide it out into PFO negative and PFO positive, this time that positive and significant relationships being driven by our PFO negative group, whereas now there's no relationship between core temp and heart rate in our PFO positive group. Moving on to a higher exercise intensity, you can see the same thing we saw pre-exercise and at 50% of VO2 max. In the whole group data, there's a positive and significant relationship between core temp and heart rate. And we divide it out to look at the effect of a PFO. You can see it's still being driven by our PFO negative group, where there's no relationship between core temp and heart rate in our PFO positive group. So what we know so far is that PFO positive men have higher esophageal temperatures at rest during exercise and during passive heating and cooling. The higher esophageal temperatures compared to PFO negative subjects seems to be dependent on the size of the PFO, so large versus small PFOs. PFO positive subjects have a blunted thermal hyperpnea, so they increase their breathing at a higher esophageal temperature, and they have a less of an increase in minute ventilation after that ventilatory threshold. PFO positive subjects maintain higher esophageal temperatures during cold exposures and start to shiver at a higher esophageal temperature compared to PFO negative subjects. And there appears to be differences in the inter-individual relationship between heart rate and core temperature at rest and during exercise between PFO positive and PFO negative individuals. So talking about some limitations of our previous study and what we're working on now. So when we were designing our next study, we knew we wanted to do a little bit of a longer exercise test. In Jim's first exercise study, the stages were only 2.5 minutes long, so not quite long enough to reach a steady state exercise. So we didn't know if maybe that was impacting some of the results that we were seeing. And our initial plan was to do 60 minutes of exercise at 60% of VO2 peak. And then through talking to people like Ollie J, we learned about the importance of doing exercise intensities as a heat production rather than a relative exercise intensity. So if we look at this table of 2 subjects from that first study, we can see that both subjects are at 50% of VO2 peak. Subject A is at 175 watts, and they're producing almost 11 watts per kilogram of body heat. Whereas subject B is at 137 watts, and they're producing almost 5 watts per kilogram of body weight. So in these subjects, we don't know whether or not we're actually seeing differences in core temperature due to differences in thermoregulation, or if the differences we're seeing are just because these people are producing different amounts of heat. So we can use these formulas and calculate metabolic rate and no external workload and no body weight and calculate how much heat people are producing per kilogram of body weight. We also have done no studies looking at women with the PFO, so we don't know if we see the same core temperature elevation in PFO positive women that we see in PFO positive men. But we do know that we see temperature shifts along with the menstrual cycle. And in the luteal phase, we have an elevation in temperature by about 0.3, 0.4 degrees Celsius due to progesterone. So we don't know if our PFO positive women do have higher temperatures, if there's an additive, if there's a synergistic or antagonistic effect with the temperature changes we see in the luteal phase. So our study we're doing right now is looking at the influence of a PFO on core temperature responses during 60 minutes of exercise at a given heat production. And we're studying both men and women in both menstrual cycle phases or in the low hormone or high hormone phases of birth control. So on the first visit to the lab in the study, we do what we just call a heat production test, followed by a VO2 peak test. And these methods came from a paper done by Ollie J and Matt Kramer. And you can, during the heat production test, we have subjects cycle at 4 different workloads for 5 minutes apiece. And we're able to find the heat productions at each of those workloads. And it gives us a regression equation that we can find our ideal workload that will elicit a heat production of about 7 watts per kilogram. So in this person, we had a target workload of 91 watts. I will say that in most subjects, we've had to adjust their target workload by about 10 to 15 watts to get the ideal heat production during the hour-long bout. During the 60-minute trial, we're measuring core temperature with a telemetric pill and esophageal temperature with an esophageal probe. There's been some idea that maybe the PFO is just a physiological hotspot, and we wanted to see if we still see these differences in core temperature in different sites in the body rather than just using esophageal probes. We're also measuring skin temperature, measuring respiratory heat loss. As Andy mentioned earlier, Caitlin's seen some differences with inflammatory markers. So we wanted to measure some of those. We're getting estimates of whole body sweat loss. And in this study, we're controlling for the amount of heat produced, the time of day, and their hydration status. So I have some results for you. I don't have any PFO positive women in our study yet. So these are all data from our PFO positive and PFO negative men. We've also had some issues within our esophageal probe system. So I only have pre-exercise data. But right now with 3 PFO positive and 4 PFO negative men, there's no difference in esophageal temperature. But we would almost expect this at this point because we only have one subject with a large PFO. So if we think about the temperature differences we're seeing really being driven by the size of the PFO, we wouldn't necessarily expect there to be large differences yet. But also, I just spent 22 slides telling you that PFO positive subjects have warmer temperatures. But right now, at a controlled heat production over 60 minutes of exercise using the telemetric pill, our PFO positive subjects are a little cooler than our PFO negative subjects. There's no stats run on this yet. We only have 5 PFO negative subjects and 6 PFO positive subjects, but it's definitely really interesting that there might be differences in temperatures between the different locations. We're also measuring skin temperature. So PFO positive subjects, skin temperatures are a little bit higher than PFO negative. But when we do the calculation and calculate mean body temperature using the telemetric pill data and the skin temperature, we can see that there's no difference in mean body temperature between these two groups. So for future studies, we'd like to look at skin blood flow responses, both in the heat and the cold, see if there's differences in sweating, both at the onset of sweating and the amount of sweat. We don't know whether or not we'd see the same responses to hot tub and cold tub exposure in PFO positive women that we saw in PFO positive men. So we'd really like to do that. And then we don't know if there's differences in metabolism or shivering or non-shivering thermogenesis. So maybe measuring things like uncoupling proteins could help us determine what some of the differences are. I'd be happy to take any questions.
Thank you for that wonderful talk, Karleigh. It's really really nice overview of the thermal physiology work that you've been doing. I guess this was a topic raised by, or a point raised by Andrew earlier, but why might PFOs kind of have different sex effects? Can you kind of give us some ideas of kind of the theory behind that, or anything that you know?
Like, why would we see differences in men, but maybe not in women?
Mm-hmm.
Oh gosh, I don't know. I'm not sure we know that in terms of thermoregulation. Yeah, that's a really good question, but yeah, I'm not sure. I don't know if Andy has any thoughts on.
Andrew, do you want to reflect back on, that was one of the points you made about kind of women seem to be, I think it was more susceptible to hypoxia.
Yeah. So I'm not muted. Yeah. So yeah. So I would say that it's possible that if the blood flowing across the PFO is what's really contributing to this to the differences in temperature, then it might be when women, if they've got a large PFO and they have an extra blood flowing across that may cause them to get hotter while they're exercising compared to a woman without. But we don't have any data to support that. We don't understand the relationship between the size and the temperature effect. It's easy to come up with a reason why the gas exchange would be different, because it's more venous blood mixing with oxygenated blood, but I don't know how this works with the temperature thing. It's still quite the mystery.
Yeah, well, I mean, that's the fun of science, finding this stuff out. We have a question from Vienna Brunt. Nice to see you again, Vienna. Nice job, Karleigh. Are PFO individuals at higher risk for heat stroke? What are your thoughts on that, and kind of the risks, I guess, both from public health and also potentially for occupational health too?
Yeah, that's a really good question, and something we've definitely been talking a lot about. I guess my answer to that would be maybe, but we also know that there are a lot of other things that contribute to a heat stroke or a heat illness besides just temperature. So whether or not someone's gotten enough sleep, if they've had a recent illness and things like that. We also know that there can be, some people could be exercising, their temperature could be above 40, whereas other people's will have a heat illness at maybe 39 degrees Celsius. So maybe, but I don't know if just the higher temperatures itself would help contribute to the differences in heat stroke or heat illness.
Thanks for that. And we have a question from Adam Park. I think it was the cold air inhalation trials that showed elevated esophageal temperatures. Would you have any predictions as to what the response would be to inhaling hot air instead of cold in PFO subjects? Would it, in essence, make it much worse if they were breathing hot air?
I guess if we think about it that if we're thinking that our PFO positive subjects are worse at dissipating heat, I'm guessing we'd see that they'd have greater increases in temperature in response to breathing hot air compared to maybe PFO subjects with a small PFO or subjects with no PFO.
Mm-hmm. Which really, again, raises back to the idea of the hyperventilation that we're seeing, or kind of the delayed hyperventilatory responses too with body temperature. And again, it'd be interesting to see, if you were kind of breathing hot air, which is certainly the case, potential case in again, occupational physiology, how that might impact anything. Today, any questions?
Yeah, thanks a lot, Stephen, for the opportunity, and Karleigh, I mean, great talk, obviously. So if I understood correctly, the most of the data that you've shown is actually indicating that PFO positive individuals, either men or women, are essentially, their basic core temperature is a bit higher, except for your latest data. Is that correct?
Yes. Yep. With everything except for telemetric pill data so far.
Yeah. Okay. So this in the end, but maybe this is an extension of Vienna's question, because this was an obvious question for me, if they're more at risk of whatever heat-related illnesses. But do you know, or I mean, is there any idea why what's the underlying mechanism of this higher body temperature at rest, for example? I mean.
Yeah, so we're in the study we're doing now, we really want to look at more get a better idea of inflammatory markers. Not that these people necessarily have a fever, but if, you know what I mean, if they have more like increased inflammation, if maybe it's increasing body temperature through that mechanism. But that's really our only idea right now, is that maybe there's just higher levels of inflammation that maybe are contributing to that. Yeah,
because it's, I mean, you know, it's really quite an important applied question in a way, you know, because if they're all the time working on a higher temperature, I mean, it must have many metabolic and other effects on the body. So yeah.
Yes, the other question, the flip side of that question, is whether it's just being regulated at a higher baseline,
right? Yeah.
Whether it is really, absolute temperature, or whether that's just, again, that person's normal baseline temperature, as opposed to being an abnormal type of temperature.
Yeah.
But I think I can diagnose that I probably do not have a PFO, given my constantly a low core temperature. Whenever my students always end up having to, like, Joan, is this probe working? But yeah, any other questions from the audience? I guess any closing words from either Andrew or Karleigh?
We got some questions, it looks like.
Okay, and there's Tony again. Preliminary data suggests Dr. J might be correct. Thoughts? I guess in terms of the watts per kilogram, like once it's normalized to that?
Yeah, I would agree so far. Maybe it doesn't really, like we just talked about, it doesn't explain the differences we're seeing at rest, but it might explain the differences we're seeing in response to exercise for sure.
He's responding no hotspot. Does that ring a bell? I'm not quite sure what that.
Oh, that we'd.
Oh, right, esophageal versus rectal.
Yeah. So definitely that the differences could be because of the esophageal probe. It's kind of a bummer that we've had some issues with our equipment, because we can't compare the people we have so far to see if, you know what I mean, if it's just maybe subjects we've studied in the past that have had those differences in esophageal temperature. But yeah, it could definitely be a location thing.
Okay. And then Adam Clark again, do you think the acute myokine inflammatory response to exercise would be more robust in PFO subjects?
I think that because the deltas that we see over the course of exercise and temperature is not different. So I think in order to know that, we would need to study temperature responses maybe after in order to get an idea and measure inflammatory markers after. But I'm guessing if these people have increases of inflammation to begin with, there's something that's process has already started. So they might have higher increases after exercise compared to PFO negative subjects.
I guess actually also to really tease out today's question and also this is another approach might be not just looking at the response during exercise, but really the recovery, right? Of, you know, again, if the idea is that with blood shunting, that there is less respiratory heat loss and less heat clearance, you would actually expect to see that more in a sense play out during the recovery, that they would have a much longer lag time of returning to baseline compared to PFO negative individuals. Any ideas or thoughts about that?
No, we're right now we're having them stay for about 10 minutes post exercise, but that's a good thought. Maybe we start keeping them a little longer to get an idea about what's happening a little bit longer post exercise.
Yeah, love to follow that up with you afterwards. But that would be an interesting kind of approach to looking at, you know, not necessarily just the onset of heat, but also really, in essence, the dissipation of heat post exercise. All right, so with that, I will just share the screen, my screen again. So again, I want to thank both Karleigh and Andrew for really enlightening, interesting talks to hopefully bring this area more to the forefront of our thoughts, and just as an interesting kind of mechanism that we can use to really explore physiology. So I will finish off again by reminding you that we are hosting ICEE live and in person September 6th to 10th here in Niagara in Canada, which is a very short hop from both Toronto and also from Buffalo. So we're pretty much right at the border. And again, you can find information for registration and also for abstract submission. It is open live now at ICEE2022.com. And again, you have the email there, ICEE2022Niagara@gmail.com. If you want to ask me any questions or anyone else in our organizing committee. Otherwise, again, I want to thank you. We will, again, have all of the talks, as always, up on the Environmental Ergonomics YouTube channel. And I hope you all have a rest of an excellent day. Thank you again very much for attending, and thank you to our speakers today.
Thank you, Stephen and Tari.
Thank you.
Yeah, thanks a lot. I mean, it was really insightful, you know, it's wow. Lots of unknowns, I guess, lots of holes in the story.
And finding more all the time, it seems.
Yeah.
So.
Like,